Hey guys, it's Nora. Today's video is going to be a really quick one about a very common problem in bioinformatics, which is I want to do the same analysis, but visualize it a few different ways. So what this can look like practically is, okay, I have to go back to R and pick a new ggplot function, and then, I may not be super comfortable with the parameters, and then it's, 20 to 30 minutes to do something simple like change a plot type or change your colors. So I want to show how to do the same thing in just a couple minutes on Pluto. So I'm going to click this Add Analysis button and start with just a summary plot of my normalized counts. As always, I'm doing this through a scientific lens, so I have a list of genes that are associated with differentiation and glial guidance, and I want to compare their expression across pre-tumorigenic astrocytes and tumor-propagating cells. So I'm going to run that analysis, and it's going to deliver a nice bar plot to me. So I'll go over to this plot tab and change just a couple features. So I'm going to change the colors to be yellow and blue. And then the next thing I'm going to do is run a quick statistical test. So I'm going to just choose one of these genes, so pick SMAD7, and run a 2-sample t-test between my groups. And then I see that the output is statistically significant, so I want to visualize that directly on my plot. Okay, so that is plot number 1. We just have a nice bar plot showing the expression of our normalized counts across the two groups. Another way that we could visualize this is through a heat map of the gene expression data. And again, I'm just going to change those colors to be yellow and blue and click Save Changes. And then it's going to update to a new plot. Okay, so that's 2 ways of doing it. And then the last way that I will show is slightly different. We're going to look at an actual differential expression, which is looking at log fold change across your samples. So we'll click Create Analysis, select our groups, which again is the astrocytes and the tumor propagating cells. Click Run Analysis. This will churn for a few seconds, and then it'll deliver a nice volcano plot showing the significantly increased genes on the right and the significantly decreased genes on the left. So again, let's just adjust those colors really quick. So I want to change the significantly upregulated genes to be cobalt blue. And I'll do that for the insignificant upregulated genes as well. And then we're also going to change the significant downregulated genes to be yellow, just stick with our color scheme. We'll do this for insignificant downregulated genes as well. Okay, and then we will make our labeled genes, which I'll show in just a second, be a nice bright green for contrast. And the last thing we want to do is actually label those specific genes that we are interested in, which is our differentiation and glial guidance genes. I've created a list that tracks those specific genes, so it's really easy to select from. Save changes, and then we can see that those genes are indeed significantly increased in the pre-tumorigenic astrocytes compared to the tumor propagating cells. So I'm just going to close out of this plot for a second and look at the whole picture. So we looked at how to analyze the same thing across a few different plots, but what did we actually learn from a science standpoint? So we see that these differentiation and glial guidance related genes are increased in the pre-tumorigenic astrocytes compared to the tumor propagating cells. So what we actually learned from a biological perspective here is that there's a downregulation of these differentiation related genes during the transition from those pre-tumorigenic astrocytes to the tumor propagating cells. And that makes sense because that transition is a hallmark of how cancer cells acquire stem cell-like properties. So those tumor propagating cells behave like cancer stem cells, they self-renew and they generate new diverse tumor cell populations. And part of that is, or how they achieve that is by suppressing those differentiation related programs. Thanks so much for joining. I hope you learned a little bit of science and also a little bit of Pluto's capabilities in this video, and I'll see you in the next one. Bye.